Dear students, welcome you all in the virtual process of learning. Today, I am Deepika Paliwal. Welcome you all on the behalf of School of Medical and Allied Sciences, Calcutta University. Today we will learn about anti-cancer agent. Anti-cancer agents are the chemical agent used for the treatment of cancer. Before we start the lecture, the student must be aware about the structure of the DNA, structure of RNA, and the process of protein synthesis. These factors are very important for today's lecture. In today's lecture, we will learn about different types of cancer, what are the different approach for treating the cancer, and what is the chemical classification of anti-cancer agent. Introduction of cancer. Cancer is a term used for the diseases in which abnormal cells divide without control and are able to invade the other tissue, means they are transferred from one organ to others. Cancer cell can spread to other parts of the body through the blood and the lymph system or the lymph node, and this process is called metastasis. Characteristic of the cancer cell. Cancer cell involves the development and reproduction of abnormal cells. Cancer cells are usually non-functional. Cancer cell growth is not subject to normal body control mechanism. Cancer cell eventually metastated to other organs via the circulatory and the lymphatic system, means they are circulated through blood and the lymph fluid. Type of tumors. Tumors are divided into two categories, benign tumors and malignant tumors. As we see in this picture, benign tumors is human friendly, whereas malignant tumor is enemy to us. So benign tumors are non-cancerous and not an immediate threat to life. Even through treatment eventually may be required for the health. Benign tumors are treated by using the treatment. Malignant tumors are tending to worsen and they lead to death. These are invasive and metastatic. Cell cycle. This diagram represents the mammalian cell cycles. This is the G0 phase. It is also known as the resting phase, where cell is not dividing. In the G1 phase, there is synthesis of the enzyme that are required for the DNA synthesis are synthesized here. In the S phase, DNA is replicated. In the G2 phase, these are the synthesis of the cellular components that are required for the mitosis or the cell division process are synthesized here. In the M phase, that is also known as the mitotic phase, where the cell divides. So this representation of the mammalian cell cycle. In each cell division cycle, chromosomes are replicated once and the DNA synthesis or the replication and segregated to create the two genetically identical daughter cells. That is also known as the mitosis or the M phase, and this event are spaced by the interval of growth and the reorganization. The gap phases between the G1 and G2 cell can stop cycling after the division, entering a state of quiescence. The treatment of cancer is basically based on this cell cycle. There are the two categories, cell cycle specific drug and cell cycle non-specific drug. Under this category, cell cycle specific drug, the drug which are acting on the specific cell or specific phase are the antimetabolite, bleomycin, antibiotics, vinca alkaloids, and etoposide. These are effective for the high growth fraction malignancies, for example, the hematological cancer or the blood cancer. Cell cycle non-specific drug. These do not act on the particular phase, but they kill or inhibit the cell growth are the alkylating agent, antibiotic, cisplatin, and nitrosourea. They are effective for both low growth friction malignancies as well as solid tumors as well as the high growth friction. Classification of anti-cancer drug. Anti-cancer drugs are categorized into many four categories that are alkylating agent, platinum coordination, antimetabolites, plant products, and the antibiotic. We will discuss these classes one by one. The first class is alkylating agent. As the name indicates, they make a covalent bond with the DNA, and the drugs comes under these categories are alkylating agents are further categorized into four categories. The first category is nitrogen mustard compounds. They are substituted of the mustard gas with the nitrogen atoms. The drugs comes under these categories are mechlorethamine, cyclophosphamide, ifosfamide, mefalon, and the chlorobenzene. These are the derivative of mustard gas. These are the structure of these drugs. The second category of alkylating agent are ethylene amines. The drugs under this category are thiotepa, hexamethylmelamine. These containing aziridine moiety in their structure. The third category is alkyl sulfonate. The drug comes under this category is busulfan. It containing the disulfate group and the butyl chain in their structure. The fourth category of the alkylating agent is nitrosourea. The drug under these categories are carmestine, lomustine, and streptomycin. They are the nitro derivative of urea. This is the structure of urea and this is the nitro derivative. So in all these structure they containing urea and nitro in their structure. The last category of alkylating agent is triazine. The drug comes under these categories are procarbazine, dacarbazine, and temozolamides. These containing three nitrogen atoms in their structures. The next category is platinum coordination complex. These are containing platinum derivative and they make a coordination bond with the compound. The drug comes under this category are cisplatin, carboplatin, and the oxaliplatin. These are the structure of these drugs. After the discussion of alkylating agent and the platinum coordination complex, third category is antimetabolites. Antimetabolites are the drug which having the same similar structure as the biological precursor of the process. These antimetabolite category are further categorized into three categories: pyrimidine analogs, purine analogs, and the folic acid antagonist. The pyridine analogs containing pyridine ring in their structure. As you can see in all structure they containing the pyrimidine ring. The drug comes under this category are 5-fluorouracil citrabine or also known as the cytosine arabinozoate. They are the sugar derivative of pyridine moiety, capecitabine, and the gemcitabine. These are the structure of derivatives. Except 5-fluorouracil, all are the sugar derivatives. The next category of antimetabolites are purine analogs. They are derivative of purine nucleotide. The drugs under this category are 6-mercaptopurine, 6-thioguanine, azathioprine, fludarabine, cladribine, and the pentostatins. 6-mercaptopurine is the derivative of sulfahydryl derivative of hypoxanthine. Thioguanine is the amino derivative of mercaptopurine. These are the structure of different drugs. The last category of antimetabolites are folic acid antagonists. drug comes under this category are methotrexate and pemetrexate and the trimetrexate. These are the derivative of folic acid and inhibit the biosynthesis of folic acid, which is important step in the synthesis of DNA. These are the structure of these drugs. The next class of anti-cancer drugs are anti-cancer antibiotics. Antibiotics are the chemical agents which are produced from the microorganism and used to kill or inhibit the growth of other microorganisms. The drugs under these categories are actinomycin, bleomycin, mitomycin C, anthracycline, which contains the drug doxorubicin, daunorubicin, idrarubicin, apirubicin, and the valirubicin, and the streptomycin. As we saw the chemistry of these antibiotics, dactinomycin containing phenoxygen ring in their structure with the peptides, whereas anthracycline containing tetracycline ring with the sugar, unusual sugar bond with glycosidic linkage. These tetracycline ring containing compounds are also known as anthracyclines. The last category of anti-cancer agents are plant products. They are derived from the plant. Examples are vincristine, vinblastine, podophyllitoxin, etoposide, camptothecin, and the paclitaxel. Phyllotoxin, paclitaxel, and etoposide are taxol derivatives, whereas vincristine and vinblastine belongs to vinca alkaloids. These are the structure of these drugs. Let me come with summary of today's lecture. In this lecture, we will learn about the cancer. Cancer is a term used for the disease in which abnormal cells without control and are able to invade the other tissues. Cancer cell can spread to other parts of the body through the blood and the lymph system, and this process is known as the metastasis. Tumors are the two types, benign tumors and malignant tumors. Benign tumors are non-cancerous and not an immediate threat to life, even though treatment eventually may be required for the health, while malignant tumors are tending to worsen and leads to death. They are invasive and metastatic. Anti-cancer drugs are the drugs which are used for the treatment of the cancer. There are main groups of anti-cancer cytotoxic drugs are alkylating agent, antimetabolites, antibiotic, and plant products. Alkylating agents are a class of drugs that are capable of forming covalent bonds with the important biomolecules, whereas in the antimetabolites compounds closely related in the structure to a cellular precursor molecule, yet these substances are capable of preventing the proper use or formation of the normal cellular product. Antibiotic class. In the antibiotic, they are used to kill or inhibit the growth of other products, whereas plant products act at the metaphase, they stop the cell division process. Let me come with the practice question which comes in our exam from this lecture. The first question is the difference between the benign and the malignant tumor, and what are the different classes of anti-cancer agent. Discuss the detailed classes of the alkylating agent and what are the different classes of antimetabolites. Dear students, if you want more information regarding this lecture, you can refer to this reference book. We will meet in the next lecture. Till then, take care. Thanks a lot.